Okay, a few announcements as we get started. I'm getting some feedback here and I don't know why it's doing it today and it wasn't doing it before. So sorry, that's going to impact the quality of the recording a little bit here. I have not, it's something, I muted the Zoom, so I don't know where that's coming from, but sorry about that. I was hoping, no, it doesn't have anything to do with the microphone. So sorry about that. I'll try to figure out what's going on there. So a week from today, we will have class like normal on Monday. Next Wednesday, I have an unavoidable thing that I have to do, and I won't be able to be here on Wednesday for lecture. I will be recording the lecture hopefully the night before. All right, so I will send out some announcements to remind you of that. I hate to disrupt the habit that you're getting into coming to class. I think it is good to actually come to class and have in-person class. I think most students benefit from that, but so again, sorry that I will have to miss next Wednesday. Also, I made a couple of errors that some of you have asked me about, and then I've been able to fix them. But in case you didn't ask me about it and you were wondering, it looks like most of you are registered for mastering now. Oh, that bugs me. It's something, that monitor, but I don't know what it is. Okay. If you are still having trouble registering, make sure you let me know. I had it accidentally set to close registration on Monday. So if some of you have tried to do it since then, you should be able to do it now. I changed that date. That was just an error made. And I also realized, I looked in today at the grade book and I thought, oh, hardly nobody has done the homework from chapter 1. Well, I accidentally made that not available until yesterday. So if you were looking for chapter 1 homework, or maybe you didn't notice that it wasn't there, but I just had set the date wrong there. So it's not a very long assignment there. You should be starting to work on that homework. Again, don't let that build up till a few days before the exam. The exam is not next Wednesday, but the week after that. So we still have a little time there, but you should be working on the chapter 1, the chapter 4, chapter 6, 11. We're going to start chapter 3 today. So there's some assignments there that you can be getting started with. Okay, all right. Any questions from last time? All right. I don't know what's going on with that. Where we finished, we pretty much finished up chapter 4. We had ended talking about some different culture medias, lots of different medias that we can use depending on what our goals are, what we're trying to learn about bacteria, or what we're trying to grow up. We talked about different kinds of microscopy. You should be familiar with those ones that we covered in class, not only what cells look like when you use that kind of microscopy, but maybe some advantage of it or disadvantages of those kinds of microscopy. And there's more examples in the book. Don't worry about some pictures I show you, like don't worry about recognizing what kind of bacteria that is. You should be looking for, the principles like, oh, it's a dark background, so it may be, dark field microscopy, for instance. We talked about classification a little bit and how you're kind of moving towards some more complex criteria for classification, like for instance, ribosomal RNA sequencing. And we went through this phylogenetic tree of life. Remember that was the Woese-Fox phylogeny. They sequenced a lot of ribosomal RNA back in the 70s to figure out this chart with evolutionary relationships. Okay, so all of these are 3 domains. You should know those domains, all these different kingdoms. You don't have to memorize those. I don't even recognize some of them, but that's sort of the correct system that we go by now. But so springboarding off of that, even though we often go by genetic analysis to help us with our taxonomy and organizing life, we still rely pretty heavily on characteristics that we can observe. Oftentimes, characteristics that are observed will, match up with genetic relationships as well. All right, so I just wanted to, into chapter 11, really we're talking more about kind of some very straightforward things that we can kind of view and understand to help us to classify organisms. I wanted to reiterate some differences here between those two domains of prokaryotes, the bacteria and the archaea. Okay, so I mentioned this on the first day. Big difference between those two. They both have cell walls, but bacteria have this substance called peptidoglycan in their cell walls. We're going to pick that apart a little bit later today when we talk about cell wall structure. But bacteria that have cell walls always have peptidoglycan in them. Where archaea have cell walls, they have something very similar to peptidoglycan that is known as pseudo-peptidoglycan. Not exactly the same thing. Okay, and of course, some eukarya have cell walls as well, but they never have peptidoglycan in them, right? Fungi, plants, algae have cell walls. Some characteristics of the archaea are more like the bacteria. Sometimes they have kind of characteristics that are more like the eukarya. And then sometimes they have something that's kind of very unique to the archaea. So for instance, if, picking out some details from here and there, your book just mentions a few things. If you look at membrane lipids, bacteria have, phospholipid membranes very similar to us, the eukarya. But archaea have a very unique kinds of membrane lipids. So for instance, they don't have phospholipids, they have actually a sugar at the head where the phosphate usually is, and then their lipids are these very unusual kind of ringed hydrocarbon chains. So they have kind of a unique cell membrane, for instance. Bacteria, we'll talk more about this much later, but the start codon for bacteria, AUG, encodes for that first amino acid in a protein. It's always formal methionine. It has this formal group attached, where the archaea, their first amino acid in all their proteins is methionine. That's more like the eukaryotes. All right, so again, sometimes they kind of borrow from the bacteria, sometimes from the eukaryotes, and sometimes they have their own things. So that's just some examples. All right, so chapter 11 goes through kind of general characteristics of prokaryotes. So again, these are things that we have traditionally used for classification. We still rely on them quite heavily. So kind of basics of bacteria here. If we look at the shape or the morphology of bacterial cells, all right, we have some terminology to describe those different shapes. A round bacteria cell is called a coccus, okay? Plural would be cocci. And a rod-shaped bacterial cell we refer to as a bacillus. Okay, if you're in lab, you'll see lots of cocci, lots of bacilli, and nothing else. Okay, just because not only are those forms tend to be the easiest to grow in lab, but we have lots of examples of ones that aren't disease causing. So that's mostly what you'll see in lab, but there are many other different morphologies of bacterial cells out there. So we have something halfway in between a cocci and a bacillus. So it's not a round, perfectly round cell, but it's not really a rod either. We call that, intuitive, a coccobacillus. Somebody told me once that was a question on the MCAT. What shape is a coccobacillus? And then there's some other really kind of bizarre things like this vibrio. That's a comma-shaped rod. Cholera, that's an infection that's caused by a Vibrio. And then these two shapes, these are our helical forms. When they move in a corkscrew. Okay, so they're a little bit harder to discern between. The sporillum is a little bit more rigid shape, and they typically have flagella out from one end or the other. You can't really see that there. We'll talk more about flagella pretty soon. But they're, again, flagella are on the outside, so we'll say external flagella. And the spirochetes here, they're a little bit more flexible when they move. Of course, they're longer, but they're just more flexible. And they have a internal flagella. Okay, that again, we'll talk a little bit more about later, but it's enclosed within a membrane and it runs down like their whole length of their body. All right, so more about telling the difference between those two on the next slide. But other morphologies, some bacteria don't really have any set shape. We call them pleomorphic. It could be that they change their shape depending on what environment they're in, or it could be that they don't have a cell wall. So they just don't really have any set shape. There is a group of bacteria that don't have a cell wall. And then we see some really bizarre things, mostly in the archaea, but some like star shaped or squares, or there's lots of other bizarre things out there that are a little bit more rare. Okay, so just coming back to those helical bacteria, these are pictures that I just grabbed from Wikipedia. Sometimes they don't cite very well what kind of microscopy they're using here. But what do you think, looking at this picture on the left, what kind of microscopy that might be? Of course, I don't give you the units. That would be even more helpful. Okay, this to me looks like it's a scanning electron micrograph. Okay, so that's up close of a sporilla morphology there. The little brown you can see are the flagella coming off the ends. They're a little shorter. This is actually the organism that causes many of our gastric ulcers, Helicobacter pylori. Okay, and then the picture on the right, this to me looks like it's dark field microscopy. And I know from I got to looking at that today, and I thought, if I didn't already know that, I might think that looked like it was fluorescent microscopy, because it's kind of hard. It almost looks like, there might be some dye on there that's making it fluoresce. But it's actually dark field. These are spirochetes, and this is taken from an infected tick. This is actually the organism Treponema pallidum. No, sorry, that was mixed up. That's the organism that causes Lyme disease, Borrelia burgdorferi. Okay. And if you want to see a really cool dark field, a video of some dark field microscopy, this is like a 25-second clip that shows some spirochetes kind of moving around. Okay, so again, corkscrew. We think that corkscrew movement helps them maybe move material that's a little bit more viscous. Okay, let's see. Okay, so check out that video. It's kind of fun to see them actually moving around. So remember, dark field microscopy, you don't have to stain them, so you can see them live. Same thing with phase contrast microscopy. Okay, another really easy thing besides morphology of cells to see is the arrangement. Cells tend to arrange in a very specific way, just depending on what kind of species they are. So it depends on the plane, how many planes in which the cell divides. Okay, so for instance, the top cell over here, that's dividing in one plane. Here we have dividing in two planes, so on and so forth. And the arrangement also depends on the separation of daughter cells. Okay, do they tend to stick together or do they tend to come apart? Okay, so again, we've got some terminology to describe our cell arrangements. If we tend to have, and this is for cocci, we tend to have cocci in pairs, then we call that diplococci. Okay. Characteristic of some organisms. If we still have division in just one plane, the same plane all the time, and all the cells tend to stick together, we tend to get chains of cocci. And this is also the prefix here is called strepto. Okay, strepto is Greek for chains. Okay, I have to figure out how to change that. All right, come back to where I was here. Okay, let me go back, share my screen again. Should be sharing my screen. Oh man, my mom, I'm just gonna hear it for me. All right. Okay. Okay, we're back. Okay, sorry about that. So streptococcus, for instance, that's round cells and chains. So you'll be working with a lot of streptococcus if you're in lab. Okay, if we have division in two different planes, then we have the arrangement known as tetrads. Okay, a group of four. And then if we have division in three different planes, we have a cube-shaped arrangement, and we have a special term for that, sarcina. Right, that's probably a term that's probably the most unfamiliar, sarcina. In practice, when you're looking at these arrangements under the microscope, because you're seeing a 2D bright field image, it's pretty hard to tell the difference whether you have a group of 4 or a group of 8. Okay, it's kind of hard to tell the difference between tetrads and sarcina. If you have a cell that can divide in many different planes and they stay stuck together, then we tend to have clusters of cells. And we have a Greek prefix that means clusters, staphy. Right? So staphylococcus, round cells in clusters. There are less, I don't know if I go a little further away. I still hear it. I don't know what's causing that. Sorry about the black background here. I know that white writing is hard to read in the room anyway here. Hopefully on your notes it's okay. A lot less choices for arrangement of bacilli because bacilli are always going to divide like this. Okay, so you're never going to have division of bacilli on the long plane like this. All right, so they only they grow longer, and then the septum grows in the center, and they divide. Okay, so oftentimes you're just going to have single bacilli. You might have pairs, diplobacilli, or chains, streptobacilli. Okay, anthrax, Bacillus anthracis is an organism that tends to form chains like that. And then sometimes though, bacilli will divide and then they'll stay attached maybe just at a little spot that's kind of off the center of the axis there. We call that snapping division. Okay, and so they tend to kind of form V's because they're kind of hinging back around on each other. And then eventually they kind of line up side to side like this. And we refer to that as palisades. Okay, when they are kind of lined up like this. Okay, so they're not dividing long ways to make palisades, but they're just kind of hinging back around on each other. All right, so we see that sometimes as well. All right, so what other features are easily kind of discernible that help us put bacteria into groups? One really easy thing is asking the question, do these organisms form endospores? Okay, and we're going to talk more about endospores, but in a nutshell, know that this is a survival mechanism. Okay, meaning it's not a reproductive. So in fungi, when they form spores, they're making hundreds of spores. It's a reproductive strategy. For bacteria, they're just making one spore from one metabolically active cell. It's just a survival mechanism. And very few bacteria have the ability to make endospores. Very few bacteria make endospores. Okay, so the very few genera that are able to do that, that's kind of something special about them, and it would make sense to put them all in the same group. So here's just an example of what cells look like when we've done a special spore stain to them. If you're in lab, you'll be doing that more towards the end of the semester. So we'll come back and talk more about that structure later. So what are some of these simple criteria that we can use? So we've talked about cell shape or arrangement. We are going to talk more about their gram reaction shortly, whether they're gram positive or gram negative. That's an easy thing to do to tell them apart. Do they make endospores? Also, let's think about their colony morphology. Okay, so think about those terms, cell and colony. Those are very different things. We see cells under the microscope. What we can see with our naked eye growing on media, those are colonies. And if you haven't worked with bacteria much, you probably don't have a good grasp of what those looks like. A lot of times it looks like just a bunch of dots on some media, but recognize that really there's a lot of different ways that colonies can look. And I don't want you to memorize all these terms. I just put this up here just so you recognize when you look at colonies on a plate, there's more than just white dots. Okay, they can have a very distinct shape to them. There's very different ways that we describe the margin or the edge of the cell. Sometimes they're actually elevated or kind of wrinkly. I guess umbonate is the correct term there. Many different sizes. They can be smooth, rough, dry looking, shiny, lots of different colors. So once you've worked with a species long enough, you really tend to get good at recognizing it just by looking at it on a plate. Okay. And of course, we're kind of bopping back and forth, bopping back to chapter 4 here, but there are lots of biochemical ways, of course, things that we use to put bacteria into group. We have differential media in lab that might tell us about some aspect of an organism. For example, does the organism use lactose? So we take some media with some lactose in it, as well as a pH indicator. And if the organism uses the lactose, then we get a pH change, we get a color change. So for instance, these two organisms are able to use lactose and this organism is not. We've got a way to catch whether there's gas produced when it uses lactose. So that's another way we can use to help discern maybe what kind of fermentation pathway it's using. So these are biochemical tests that help us learn about the organism. Here's a test that tells us if hydrogen sulfide is produced as a result of metabolism. We have tests that tell us if the organism can make a particular enzyme. There's a lot of variation there in the bacterial world. You'll do a test in lab to see if organisms are sensitive or resistant to certain antibiotics. So this is all information that we can learn. And there's lots of other even more complex criteria, again, like the ribosomal RNA sequencing, maybe some serological analysis, you know, some work with antibodies to get really specific information about, you know, types of proteins and things like that. So lots of other things that we can use. And if you want kind of the authoritative, you know, kind of best guess of scientists, like what we think about organisms, all their characteristics, there is one source to go to, and that's called Burgess' Manual. So we have a old copy of this in the lab, but you can look up, you know, any organism that you want and find out all the characteristics that we know about it. Okay, so there's about 5 volumes of this. I think you can find it online as well. So there is kind of one authoritative source there. Okay, so consensus of experts, that's what I was trying, the word I was trying to get to. Okay, so remember taxonomy, again, we're learning new things all the time, and that's always going to be changing. All right, let's move on to chapter 3. All right, we will be covering all of chapter 3 in pretty good detail. This is where we're talking about cell structures. All right, so some of this should be review. When we get talking about eukaryotic cells, a lot of those organelles, I'm kind of assume that you kind of know the basic idea behind those. We're going to skip through that pretty quickly. Assume that you really know the difference already between a prokaryotic and eukaryotic cell. We're going to review that. We're going to review the differences between cell walls, cell membranes, flagella. That's kind of an interesting one. Both some eukaryotic cells, some prokaryotic cells have flagella, but their structure is very different. So we'll be comparing that. And again, we'll just look briefly at some organelles. All right, so we're going to start with the prokaryotic cell and kind of work our way from the outside in, and then we'll do the same thing with eukaryotes. All right, so for review, what is a prokaryotic cell? Okay, again, hopefully this is review. This includes the bacteria and archaea domain. Okay, and the main thing that we think of when we're distinguishing a prokaryote from a eukaryote is that prokaryotes do not have a membrane-bound nucleus. They kind of have one spot where they keep their DNA, but it's not enclosed in a membrane. Okay, and of course they don't have membrane-bound organelles either. Okay, typically they're going to be smaller. Okay, typically less than or equal to 1 micron in diameter. Okay, so looking at the units here on these two pictures, here's a scanning electron micrograph of some bacteria cell. I forget what kind it is. All right, look at the units, about 1 micron, that's about 1 cell. Here's the units from light microscopy from some protozoa, okay, 40 microns. So eukaryotes are going to be typically a lot bigger. Okay. All right, so likewise, eukaryotes, they have a nucleus, membrane-bound organelles. Okay, they are going to be much bigger. So we're going to say between 10 microns to about 100 microns in diameter. So usually at least 10 times bigger than any prokaryotic cell. And this includes the algae, the fungi, plants, animals, and protozoa. So, right, we've left out viruses. Remember, they are not prokaryotes or eukaryotes. They really don't have a cellular structure. They're acellular, right? And we'll talk more about viruses later. Okay, sometimes students get mixed up when I ask them the difference between a prokaryote and a eukaryotic cell. Sometimes instead they tell me the difference between a plant and animal cell. Okay, so just like those are different comparisons there. All right, so if we start on the outside and move our way in, usually the first thing that we're going to come to is the glycocalyx of a bacterial cell. Okay, so this is a slimy substance secreted by the cell. So it surrounds the outside of the cell, typically composed of polysaccharides. Well, there's some exceptions there. There's some bacteria that have more of a protein glycocalyx, but more often it's some kind of carbohydrate polysaccharide. Okay, let's see. Okay, most bacteria produce a glycocalyx. Okay, so it's very common. All right, we didn't mention this when we talked about classification because if most organisms have them, then it's not all that useful. Okay, so very common in the bacterial world for an organism to make some kind of a glycocalyx. Okay, it's going to be considered either a slime layer or a true capsule. So that term capsule is maybe one that's a little bit familiar. If the glycocalyx is really quite rigid and tightly bound, we would consider it a capsule. If it's a little bit more loosely organized, we might consider it more of a slime layer. So it's kind of a continuum there. Whether it's a slime layer or a capsule, the function is the same. Three major functions of the glycocalyx. One is attachment, okay, maybe either to the host or to other cells. Okay, so a bacteria that is trying to colonize our mouth, if it can make a nice slimy capsule, it's going to be able to adhere to our teeth and not get washed down into the digestive system. So that's a really important function there. Cells that can attach to each other might be able to form biofilms, and that can be an advantage sometimes. Okay, resistance to drying out, to desiccation. Okay, so a cell that can make some kind of glycocalyx is not going to dry out as quickly. Maybe one that's coughed out from our lungs that has that capsule or slime layer around it. It's going to be viable for longer on some surface. It's not going to dry out as quickly. Okay. It's also going to give the cells an advantage. We'll say more resistant to phagocytosis. By white blood cells. Okay, so a pathogen like a bacteria, maybe it's invading our lungs. We'll talk about this much more when we get to our immunology section, but that gives those organisms a big survival advantage. It's going to be harder for our non-specific immune system, like our macrophages, our neutrophils, to engulf that pathogen if it's got a nice capsule around it. Okay, it's going to be able to resist that for a little bit longer. All right, so here's a comparison, a couple pictures from your book of a true capsule and an organism that's considered just to have a slime layer. Again, this is a continuum here. Sometimes it's hard to, or not really worthwhile labeling one or the other. But the true capsule here, it's really tightly bound. Again, this is a cross section of a cell, so that's a transmission electron micrograph. It's been artificially colored. Again, the orange part here is the glycocalyx. And another transmission electron micrograph, you can see here, even though this glycocalyx is pretty thick, it's just a little bit more loosely organized, so perhaps that would be more of a slime layer. Okay, if you are in lab, you're going to get a chance to do a capsule stain next week. It's difficult to actually stain the capsule itself because it's made of sugars, and we don't have dyes that really are going to adhere to a sugar because it's not charged. So typically we try to stain the cell wall as well as the background. And then all this unstained portion, that's a big thick capsule around those cells. Okay, so probably what you stain in class won't be quite that satisfying, but hopefully for some cells, you'll see this kind of big halo around it, and other cells that don't make as big a capsule, you'll see less of one. So that's just a picture from your book that I threw in there. All right, so the next structure that we would come to in prokaryotes is the flagella. We consider that outside of the cell still because it's not enclosed in a membrane. All right, so the main function of flagella functions in motility. So it enables the organism to move in a directed way towards some attractant. So you could say maybe chemotaxis or phototaxis if it was moving towards some light source, if it was photosynthetic. Okay, some bacteria have flagella, some don't. So if a bacteria doesn't have flagella, then it's considered non-motile, and it just kind of has to flow wherever the medium takes it. Okay. So we could break the structure of a bacterial flagella down into three things. We'll look at this more closely with the graphic. The next slide, there's a filament portion here. So that's, the long portion here. That is made of a single protein, flagellin. Let me spell that right. Okay, so that's kind of nice. That's kind of intuitive. Many subunits of flagellin protein. Okay, that is attached to a hook, which you can't really see that in this picture. I'll show that to you in the next one. And then it's attached or anchored into the cell membrane with the basal body. All right, so the graphic in your book, if you've printed this out small, those words are going to be kind of hard to see, and hear as well. But they give you two different diagrams. So this one at the top is just showing how that flagella is anchored into a gram-positive organism. Then we're going to talk about that cell wall in just a minute. And the one on the bottom here is a gram-negative organism. So you can see the cell wall here is a little bit more complicated for the gram-negative. That just means there's more of these rings here in the basal body to kind of anchor it in. Okay, I'm not so interested really in the structure of the basal body. Of course, here's the filament here, and then there's this hook portion. It's kind of curved. It kind of, it's just the change from the filament as to where it's anchored in with the basal body. But because it is kind of curved, that kind of reminds us of the motion of bacterial flagella. Okay, they rotate. So flagella in bacteria, they're always going to spin around. And that's different than eukaryotic flagella. Eukaryotic flagella move in more of a whip-like fashion. Okay, so they're always going to rotate to produce that motion. So it moves by rotation. All right, and we have different ways that we describe the arrangements of flagella. There's a lot of variety. Like I said, some cells don't have flagella. The ones that do, there's lots of different possibilities here. So some cells have paratrichous flagella. Okay, that means flagella all over their cell. Okay, so all around, they're kind of light here. You can't see them very well in that picture, but there's flagella all around. Okay, and that's actually a really common arrangement. E. coli have paratrichous flagella. Okay, that prefix peri means around. You know what the perimeter of a room is? That's the measurements all the way around the room. So that prefix peri means around. Okay, in the second picture here, it shows a single polar flagellum. So polar just means that it's at one end or the other. You would never have a flagella if it has one just sticking out the side like that. Okay, forget that. All right, so we're going to say monotrichous, one flagella. Again, it's always going to be at one end or the other. I don't believe there are any round cells, cockeye-shaped cells that have flagella. Okay, I think this is other shapes. Okay, let's see, lophotrichous. Okay, that is a tuft of polar flagella. Okay, so that prefix lopho means tuft. All right, so tuft of flagella just means kind of a group at one or both ends. Okay, that's another fairly common one. And then lastly, they don't show a picture of this, but amphitrichous, that means there's a flagella at both ends. Okay, a single flagella at both ends. All right, so that prefix amphi means both. Okay, you know what an amphibian is. It's an animal that lives in both water and land. All right, so amphitrichous flagella at both ends. Okay, and there's one other kind of flagellar arrangement, and that's an axial filament. So we mentioned that before. That's the kind of flagella that our spirochetes have. Okay, so that's enclosed in a membrane where the other flagella are not. Okay, and so this picture here shows that the flagella is actually attached at one end and the other of the body, and then it kind of moves around. Kind of trying to wrap my mind around exactly how that would look, but the flagella like moves around the rest of the cell. There's some little endoflagella, and there are many tiny endoflagella that all move apparently to bring about the whole corkscrew motion of the cell. Okay, so here's an example of another spirochete. We talked about the one that causes Lyme disease. This is the one that causes syphilis, treponema pallidum. Okay, all right. The motion of flagella is kind of interesting. So it depends on the arrangement as to what kind of motion it causes. If you've just got one flagella, it moves one direction, the cell goes one way. If it moves the other direction, the cell moves back the other way. So that's pretty straightforward. But what if you've got a cell that has paratrichous flagella all over the body? It gets a little bit more interesting. So with the paratrichous, if the flagella move counterclockwise, then they all will move together. Okay, so they all move together and you get what we call a run. Okay, so right here, It's moving, they're all moving counterclockwise together, and the cell is moving in, a very concerted direction. Okay, if the flagella move clockwise, all the flagella kind of are doing that their own way. So that produces a tumble. All right, so here they're moving clockwise, tumbling around. And if the goal is to move towards some attractant, maybe that's some nutrient, this is kind of a, oh, let's try this way. Oh, nope, that's not working. Let's try this way. Oh, that's not working. So it's a series of runs and tumbles, and eventually it will zigzag its way towards the attractant. So there's a nice animation of this that you'll do as part of your homework. I always had trouble looking at a sketch like this and understanding which way is clockwise and which way is counterclockwise. Like if I'm looking at the face of a clock, I understand that, but as soon as you like turn that a different way, my mind can't wrap around that. And it dawned on me this week, this is why I was never able to learn how all those algorithms to solve a Rubik's Cube. My son was trying to teach me how to do that, and I couldn't keep straight which way was clockwise and counterclockwise. So don't worry about that if that's not clear to you either. Again, watch those little videos, and just the point is that it causes runs and tumbles. Okay, and I put some links to some more videos. It'd be more fun to watch these together, but I can't easily call up internet sites the way I'm presenting here. This is a little short cartoon, like 25 seconds of runs and tumbles. It's pretty good. And these two are just wet mounts from light microscopy. I have done this in lab. It's kind of hard to get the contrast just right to see it, but these are really well done, and I tried to kind of narrow it down the parts that I thought was really good. You can see these little tiny cells zipping all over the place, and it's really kind of cool. So take a look at one of those, and you'll appreciate bacterial motility a little bit better. Okay, so a couple of other structures that we see on some bacteria, much more common on gram-negative. I think they have found a few exceptions to that now. So, it's almost always all or not all or none in biology, but mostly gram-negatives that you will see either fimbriae or pili. Okay, and these are very related because they have the same composition. It's the protein pilin. Okay, so because they have the same composition, sometimes we use these two terms interchangeably. We often call these appendages fimbriae if there are lots of them. So in this picture, all these short hair-like appendages are the fimbriae. The long ones are some flagella. If there's lots of them, and lost my train of thought. If they're fairly short, we tend to call them fimbriae. An organism would use fimbriae primarily for attachment. Okay, so for instance, E. coli has lots of fimbriae that it uses to attach itself to endothelial cells of our large intestine. Okay, otherwise, if they didn't have fimbriae, they would just get washed out. Okay, so for a number of bacteria, those are really important to attach to its type of host cell. Pili, we typically call this appendage pili if there's only one or two on a cell, and they're typically a little bit longer than fimbriae. The function here is not to attach to another cell per se, but they are important in conjugation. And we're going to talk more about conjugation later. That's a type of horizontal gene transfer, right? So vertical gene transfer is from a mother cell to a daughter cell to a different generation. horizontal transfers within cells of the same generation that are living at the same time. They will extend a pilus. And we used to think that DNA was transferred through the pilus, but now we know that that just extends to another cell and then retracts to pull the cells close to each other, and a pore opens up between them, and they can exchange genetic material that way. So we'll talk more about that. Okay, but again, sometimes, you know, it's kind of, well, do you call it a fimbriae or a pili? Sometimes we can argue about that. The newest edition of your book has fixed this, but this is, it's labeled as a TEM, right? That is very clearly, to me, not a cross section. Okay, we're seeing the outside of cells. Okay, this should be a scanning electron micrograph and not a TEM. Again, I think that's fixed in the 6th edition now, but okay. All right, so then we come to the bacterial cell wall. Okay, so we're spending a lot of time kind of outside of the cell and the cell wall, but don't worry, there's not a whole lot actually inside the cell to be going through, so don't worry. All right, so bacterial cell wall, what's the function of that? Does impart the shape to a cell, and very important for strength. Okay, if the integrity of the cell wall is compromised, then it's much more likely to burst due to changes in solute concentration. All right, so if we had water rushing to the cell, it would be much more likely to lyse. So it's very important for a cell to keep that integrity of that cell wall. All right, so what all bacterial cell walls have in common? Number one is that structure, peptidoglycan, that I've mentioned a few times. Okay, so all have a cell wall, it's got some peptidoglycan in it. All right, so if we look at this word, that glycan, that should make you think sugar. All right, so actually it's composed of 2 sugars. Okay, N-acetylglucosamine. Okay, we usually abbreviate that NAG, and N-acetylmuramic acid. Okay, NAM. All right, so here's 2 sugar subunits alternating. So NAM, NAG. All right, we'll look at the structure just shortly. So repeating sugars, and then this portion of the word peptido, that should make you think peptide. There are also some amino acid cross bridges. Okay. All right, so it's a very rigid, strong structure. Okay. So if we look at those sugars, I don't think I posted this because, right, I don't need you to memorize this. If you've had biochem, you're familiar with structures of sugar, with the structure of glucose. You can see that the NAG and the NAM, they're both very similar to the structure of glucose. There's only, let's see, I guess, you know, a couple parts here that slight variation. And of course, NAG and NAM, they're very similar in structure as well. So these are sugars. And here's the, this is from your textbook. Just so you have kind of idea, okay, those sugars are alternating, and then these little circles here, we've got some amino acid bridges between the columns as well as the row. So the analogy that always comes to my mind is a chain link fence. Okay, it's a pretty rigid, tough structure. It's fairly permeable. So as long as something is small enough to fit through there, it will go right through, right? It's a cell membrane's job to only let certain things in or out. But the cell membrane is kind of dumb that way. As long as something's small enough, it can go right through. But it does impart strength. Okay, I want to talk briefly. Your book does not mention anything about what happens when the cell grows. Okay, but I want to touch on this because this becomes important later on when we're talking about antibiotics and understanding the effect that many antibiotics have on cell wall. Okay, so in order for the cell to grow, they have to expand that peptidoglycan shell. All right, so the cell gets bigger, they've got to add on, make their cell wall bigger. And they have to do that kind of in a very careful, orchestrated way. Okay, certain enzymes called autolysins are going to break the bonds, actually glycosidic bonds between the sugars in the NAM and the NAG, and the amino acid cross bridges. Okay, so that's one enzyme. The autolysins will cut those bonds, and then new subunits will be brought in, and another enzyme, transpeptidases, or enzymes called transpeptidases, are going to reseal the bonds in the amino acid cross bridges. Okay, it's actually a different enzyme that will seal up the bonds between the sugars, but I just kind of left that out. It's the transpeptidases that we're really interested in here because those enzymes are what a whole class of antibiotics called beta-lactam antibiotics bind to. And they keep those enzymes from doing their jobs. So if you've got too much cutting going on with the autolysins and not enough resealing, okay, you're gonna lose the integrity of that cell wall. You're gonna start to get some holes in it, and then the cell is going to likely burst due to water coming in when it's in a hypotonic environment. Okay. So the orchestration of the autolysins and the transpeptidases, you got to have kind of the same amount of each of them, same amount of resealing as you have as of the cuts. All right, I'm going to come back to that video and talk about that in a second. But here's the best picture I could find of this. Of course, the sugar subunits are going to be synthesized inside of the cell, and then they need to be transferred through the plasma membrane. There's special channels for that to happen. The autolysins are going to cut these bonds between the NAN and NAG. The subunits are stuck in there, the new ones, so that it can grow. And again, certain enzymes reseal these glycosidic linkages, and the transpeptidases have to reseal those amino acid cross bridges, right? So again, very, very carefully orchestrated. If there is some antibiotic that's binding to those transpeptidases, again, then the cell is likely to lyse. And this is a really nice time-lapse video. It's just real short, but it shows some cells actually just bursting. Okay, so that's kind of interesting to look at. All right, so what is the difference between a gram-positive and a gram-negative cell? It comes down to differences in their cell wall. Okay, the gram-positive cell wall is somewhere between 40 and 80% peptidoglycan by weight. I see lots of different estimates of that percentage in the literature, a big range there, but a good part of the cell wall of a gram-positive is basically made of peptidoglycan. So a little mnemonic that helps me remember that, okay, gram-positive cell wall, lots of peptidoglycan. Gram-negative cell wall, very little peptidoglycan. Okay, so a gram-negative, I'm getting ahead of myself a little bit, is only about 10% peptidoglycan. Okay, so it's a big difference in the amount of peptidoglycan, and that actually dictates how well, during our staining procedure, the stain sticks a lot better to the gram-positive, doesn't get washed out as easily. Okay, so that's why the gram-negatives will stain a different color as the gram-positives. Okay, let's see. Let me go through that just real quickly, because if you're not in lab, the gram staining procedure, this might be the only time that you have contact with it. If you are in lab, you're gonna get to do this next week. When you gram stain your cells, you put them on a slide, you usually heat them a little bit so that they stick to the slide. They don't get washed off during the staining procedure. You add crystal violet, that's your first dye. Leave it on there a little bit, wash it off. We add a little bit of iodine, grams iodine. We call that a mordant. It's just something that makes the crystal violet really adhere to that cell wall really well. And then the third step is our decolorization step. We decolorize with acetone dissolved in alcohol. Okay, again, that decolorizes. We only leave that on a few seconds, but because the gram negatives have so little peptidoglycan, that crystal violet washes out right away. The gram positives will hold on to it longer. All right, so you just do that for a few seconds, you rinse it off, and then because we still want to see the gram negative cells, we have to add a counter stain, safranin. Okay, and then our gram negative cells will stain red from that safranin, and our gram positives will stain purple. They hold on to that crystal violet. Okay, so I did not post this. I just threw this up here today. It's a little bit fuzzy. That was a picture I took. But here are purple cells here. Those are gram positives. And here's some gram negatives mixed in, the red ones. Okay, sometimes students want to say, oh, round cells are always gram positives and bacilli are always gram negative. And that is not the case. So the gram reaction doesn't have anything to do with the morphology of the cell. Okay, we have lots of gram negative cocci, we have lots of gram positive rods. So a couple of other things that are unique to the gram positive cell wall. Actually, let me just write that on the next page here where we're labeling everything. This pinkish part here, that's the peptidoglycan. This is a figure from your book that we're gonna add the labeling to. So that big fat pinkish part, that's peptidoglycan. These long blue, I don't know, that's supposed to be lipoteichoic acids. Okay, so these long blue strings that kind of attach the peptidoglycan into the plasma membrane, or we'll call that the cell membrane. Okay, and the little blue spots there that aren't attached into the cell membrane, we just refer to those as teichoic acids. It's not abundantly clear the purpose of lipoteichoic acids and teichoic acids. We know that the lipoteichoic acids, anchor the cell wall into the cell membrane. Teichoic acids, we think that they have some role in cell division. Again, it's not terribly clear what that is, and they have some role in determining the shape of the cell. Okay. All right, so that's pretty much it. There's not anything terribly complicated about a gram-positive cell wall. Okay, it gets a little bit more complicated looking at the gram-negatives. They have an extra layer in there. Okay, so like I mentioned before, they have a thin layer of peptidoglycan, only about 10% of the cell. They also have an outer membrane. And what's composed of that composition of that outer membrane? They have a phospholipid bilayer. Can't spell, that's an I. Okay, so it's similar in some respects to the plasma membrane. There's also some proteins stuck in there. There are also sticking out on the very outside is a structure we call lipopolysaccharides. We abbreviate that LPS. Okay, and I'm going to show you a picture of this in a minute, but that's composed of several polysaccharides. Okay, so 2 polysaccharides and lipid A. Lipid A also goes by another name. We also call that endotoxin. And I know that doesn't necessarily make sense. This is on the very outside of the cell. Endo makes it sound like it's something that's within the cell. We originally thought it was inside the cell, so it's kind of a misnomer. All right. We'll get more about that in a minute. The other structure that is unique to gram-negative, they have some porins in their outer membrane. Okay, these are just protein channels, okay, for transport. Of things inside and outside of the cell. All right, so if we look at our picture here from our book of a gram-negative cell wall. Okay, so here this pinkish patty in the middle reminds me of a like frozen Wendy's hamburger or something like that. That's the peptidoglycan layer, right? You've got some periplasmic space, right below that and right above that. Of course, here's the cell membrane underneath that. And then outside here, this is what we call the outer membrane. Okay, these kind of stringy things standing up from that, that's the lipopolysaccharide, the LPS. I'm going to show you a close-up of that in a minute. And this other structure over here that kind of looks like a couple of, paper towel rolls or something, that is a porin, right? So very easy to see how that might be something for transport to allow things to, like nutrients, to come through that outer membrane. Okay. All right, the next, and there's some other, proteins in here. There's lots of proteins in membranes that are integral proteins. There's all sorts of different functions there. So we won't go into that. This is another Pearson graphic that I like, especially, it draws, I like to see different ways of representing something. So we've got the cell membrane here, the peptidoglycans drawn a little bit differently there. And then the outer membrane on the top here. And I like this picture because it shows a little bit close up the parts of that lipopolysaccharide. All right, so the stringy part that sticks up, those are a couple of different polysaccharides, so sugars that are extending out in the medium around the cell. And then that lipid A is actually right down in there, embedded into that outer membrane. And again, lipid A, also known as endotoxin. Okay, that is, let me just write endotoxin as well. That is going to be released when the cell wall breaks up. And when does the cell wall disintegrate a little bit? Mostly when the cell dies. Okay, so released when the cell dies. If you ever see some bacterial cells under the microscope where the cell wall is kind of mottled, Okay, that cell's probably dead. Okay, the cell wall is starting to break up. It didn't take up the dye very well. To some extent, a little bit of this lipid A is going to be released if the cell divides. Okay, so when the cell divides, the cell wall gets disrupted just a little bit. It might have a small amount of lipid A release. Okay, and this is important because if this is a bacteria that's in our bodies and the cell dies and releases endotoxin, lipid A, that's going to cause inflammation in our body. So our blood vessels are going to dilate a little bit. They get a little leaky. We get cells being, cells moving around out of the bloodstream into the tissues. Inflammation, fever, some kind of low-grade effects. Okay, so I will come back to this when we talk more about how strategies that bacteria have for causing disease in our bodies. But we have to think about this when we're treating a gram-negative infection in our bodies, because if we take an antibiotic that's going to kill all those gram-negatives at once, a little bit of inflammation is not a problem. You get a lot of it, it could be a big problem. So we kind of have to be aware of that when we're treating gram-negative infections. Sometimes it's best just to let it run its course rather than trying to take something to kill all of it at once. Okay, and then just again, I did not cite this, and then I couldn't go back and figure out where I got this picture, but it's just another way to schematically represent a gram-positive cell wall. On the top here, they've drawn the peptidoglycan as these kind of purple lines, teichoic acids as just kind of those yellowish or brownish lines. And then compared with the gram-negative, again, here's some porins, only a little bit of peptidoglycan there. Okay, and I particularly, I'm getting ahead of myself. I want to talk just for a minute about acid-fast cell walls. This is really a third kind of cell wall that's unique to one genera, the Mycobacterium. Okay, and there are a few other genera that are considered kind of partially acid-fast, but it's mostly the Mycobacterium. I'll explain why we call it acid-fast in a minute. It has to do with the staining procedure. But what's really special about these cell walls, they have peptidoglycan. They also have some mycolic acid in the cell wall. And there's some other unique things about it as well, but that mycolic acid imparts it's a long chain fatty acid. So it basically makes the cell wall kind of waxy. All right, so very hydrophobic. We have a hard time staining these cells because our dyes that are based in water just get repelled by the cell wall. All right, so that gives these cells some special properties. They're very environmentally tough, meaning they can withstand like disinfectants, and they're just harder to kill in general. You have a lot of high environmental stability. Okay, and they do stain weakly gram-positive. So if we had to call them either gram-positive or gram-negative, we would probably call them gram-positive. They don't have an outer membrane, so that's another reason. But really, they kind of considered a class of their own. That name acid-fast, and I'm not going to worry about, you're going to get to do this if you're in lab. The acid-fast cells here stain pink. And if they're non-acid-fast, they stain blue. But a cell, with this procedure, we actually decolorize them with acid dissolved in alcohol. So if the cell retains that primary dye, it holds fast to the primary dye, even when it's washed in acid, then it's considered acid-fast. Okay, and there's some very famous organisms that are Mycobacterium. The one that causes tuberculosis is probably the most famous one, so it's been pretty well studied. Okay, so if we compare those three kinds of cell walls, I love this graphic to compare a gram-positive. We didn't really talk about this lipoprotein there. But again, the red is the peptidoglycan. So we've got peptidoglycan in both gram-positive and gram-negative, and the mycobacterium, which are the acid fast, you know, the LPS, the porin. I'm mostly concentrated here on the mycolic acid. Don't worry about these other parts that are special about the mycobacteria. But something that I'll leave you with, and we will start with this on Monday, think about which one would be the hardest to kill. If you had some antibiotics and you know, had three different kinds of organisms, rank them in order of the hardest to kill with antibiotics to easiest. Okay, and we'll see how you do with that. We'll pick up there on Monday. Have a good weekend.